Thank you for the nice introduction. I'm indeed Luba. I do my PhD in Helmholtz, Munich, Friedrich-Alexander University, and TUM, under supervision of Paulo Casale and Bjorn Eskofier. And today I'm going to share our latest work on how foundational models can be used for genetic analysis, in particular for analysis of ocular diseases. In Germany, more than 4 million people over the age of 50 are suffering from vision loss. And this age-related vision loss can result from damage of different parts of the eye. In particular, if you have degeneration of the lens, that leads to cataract, degeneration of the optic nerve leads to glaucoma, and degeneration of macula leads to age-related macular degeneration. And although these diseases are quite prevalent, the exact mechanisms behind them are still not known, and hence there is no effective treatment that would allow you to stop the progression of the disease completely. And one of the ways to study the underlying mechanisms is through genetic association studies, where we aim to find genetic variation which affects the risk of the disease. The procedure here is quite straightforward. We collect genetic variation data from hundreds of thousands of individuals with the disease, for example, glaucoma, and without. And then variant by variant, we test for association with the trait. One of the popular approaches is logistic regression, where we have outcome as whether the person has the disease, and predictors is genotype at a given variant, corrected for covariates such as sex and age. And then for each variant, we test for beta, the genetic effect, to be statistically different from 0. And the difficulties here, of course, is scale. We do millions of statistical tests for millions of variants. One of the ways to visualize the results is through Manhattan plot, where on the x-axis we have the position of the variant, and on the y we have the strength of association with the trait. And this is the example from genetic analysis of glaucoma published in 2014. But apart from binary traits for diseases, researchers have boosted the analysis by using continuous imaging traits related to the disease. For example, glaucoma is characterized by an increased cup-to-disc ratio, which is a measurement done from a fundus image. And by doing genetic analysis of this trait, researchers have identified new 9 genetic loci for glaucoma. But these single measurements from an image capture only part of the story. The full morphology of the eye is rich in information and also multidimensional. So how can we use the full image to derive new genetic loci specific for pathology? And of course, foundational models are a great opportunity here because they allow you to capture rich and high-dimensional representation of the image in an unbiased way. In ophthalmology, a great example is RedFound. It is a model that was pre-trained on almost 1 million fundus images, with encoder and decoder parts being represented with visual transformers. And authors have also demonstrated that taking the encoder and fine-tuning it to many downstream tasks in diagnosis, one can outperform pretty much all of the baseline models. But the utility of the model for genetic analysis was not demonstrated before. So in current work, we have developed a method that would allow you to do the genetic analysis of embeddings of foundational models, for example, RedFound, and we have evaluated the utility of this analysis to understand the disease. This is how we did it in a little bit more details. We took fundus images from UK Biobank and used RedFound encoder and fine-tuned it to H by using Rankin contrast loss. The exact loss is presented here, but intuitively the representations of people that are close in age are being pushed together, and for people with larger age difference, we push the representations apart. So using this fine-tuned red-found encoder, we have derived age-tuned embeddings that, as you can see on this UMAP, are roughly ranked in latent space according to the person's age. And then we use these age-tuned embeddings in genetic analysis and also in the explainability part. Great. So for genetic analysis part, we have the outcome, our age-tuned embeddings, and we model it as a multivariate linear model of covariates, such as sex, age, and populational structure, and genetic variants. And then for each variant, we test whether the genetic effects betas across the dimensions of the embedding, whether any of them is significantly different from 0. And in this way, we have identified 178 genetic loci, which fall in genes of melanin synthesis, oxidative stress, and inflammations, which are processes that are known to play a role in eye aging. And also 27 of these variants were associated with eye diseases in a separate cohort of FinGen. And then we trained a separate conditional GAN neural network to visualize the effects of these variants. So as discussed before, we used fine-tuned red-found to get the age-tuned embeddings, and now we want to train a generator that, given these age-tuned embeddings and Gaussian noise, will reconstruct the realistically looking fundus image given the embedding. And in GAN-style training, we trained this together with discriminator that learns to identify which image is real and which is fake, also given the embedding. So training these two together, our generative decoder learns to invert the encoding procedure, and then we can use it to visualize the latent space. And we have first validated whether this conditional GAN model can visualize eye aging. For this, we fixed the Gaussian noise. We took mean embedding from 5% of people that were predicted to be the youngest, reconstructed the image. Took mean embedding from 5% of people that were predicted to be the oldest, also reconstructed the image with the same noise. And this way, the difference between the images basically shows age-related changes of this potential eye, such as vessel narrowing, blurring, and shadowing. So we can say that our embedding captures these symptoms, and also conditional GAN can visualize them. And exactly the same procedure we have applied to visualize the effects of genetic variants. First, we have assessed whether red-found can recover known genetic drivers of eye aging. If we zoom on chromosome 1 here, we can see that there is a variant that colocalizes with FinGen AMD outcome statistics and falls into a gene called complement factor H. And this is a well-known gene that plays a role in inflammation and AMD development. And indeed, the reconstructed image of predicted minor allele carriers, it looks visibly older than the left one. I'm not sure if you really can see it, but you have to trust me on that. Apart from that, red-found also have identified novel risks loci. For example, the loci here in gene KCNQ5, it was associated with age-tuned embeddings, but it didn't reach the genome-wide statistical significance in FinGen. However, it is located in a gene that is known to play a role in maintaining electrophysiological stability and electrophysiological stability of retinal neurons. And also the reconstructed images from the predicted minor allele carriers looked visibly older. Other analyses that we have performed included rare variant analysis, where we have identified 7 genes with rare coding variants associated with eye aging, and we compared the H2 red found with alternative feature extracting approaches. And next steps will include molecular and in vivo validation in collaboration with Harvard Medical School and German Mouse Clinic. Some of the take-home messages that I would like to point out is that foundational models can be used for scientific discovery, not only for diagnosis. We showed this with retinal diseases and red found, and this work required deep interdisciplinary collaborations with combining AI, human genetics, genomics, and in vivo phenotyping, which brings me to the next slide. I would like to thank all of my co-authors and supporters of this work. It wouldn't be possible without them. And you for your attention. Thank you very much.
Thank you. Very interesting talk. Do we have questions? Then let me start while they still digest. How difficult would it be? So the method you showed is fantastic in the way it works. How difficult would it be to adapt this to other applications in biomedical imaging, say, for example, progression of cancer from MRI images?
So cancer might be a little bit more difficult because it's about mutations that happen only in part of your body, and then you need to have information specifically from the cells of this cancer type, right? But there you can talk to Shubham, who is right there. He does approximately that. Yeah, so we also, in our lab, we work with brain images. So I think the general approach of extracting the embedding and then doing genomic association studies on the embedding, it can be used widely on wide of tissues and images.
But you would have to adapt the embedding.
Yeah,
okay.
Yeah. So here I use a particular foundational model that was pre-trained for eyes. Then you need different approaches for different images.
Okay, thank you. And we have one more question there.
Hi. So it's almost the question that I was asked earlier. I work with Alzheimer's, and I have seen work which kind of does the same, that they say that they can detect Alzheimer's from eye scans.
Yeah.
Are the foundational models which are out there already able to do things like that?
So, yeah, so red found that we use here in the initial paper, it was demonstrated that they also can predict Alzheimer from the eye image with quite low. The accuracy there is not in particular high. Here we look for genetic drivers of eye aging, not necessarily diagnosing any kind of disease.
But your methods were incredible anyways. Thank you for the talk.
Thank you.
Yeah.
Let me see. Yeah, we have time for one more question.
Fantastic. Yeah, thanks, Luba, for the talk. Very, very nice. I was wondering, what are your thoughts about applying this to other kinds of like high-dimensional data, single-cell data, or something like that?
Yeah. So right now we use tools and single-cell data as well as expression data to validate the loci that we have identified in genomics data. But I think, in general, if your data set allows, you can also do something very similar with, for example, expression data, right? So, yeah, this design was partially also inspired by the data that we had from UK Biobank in the beginning, but depending on the data set, you can look for many different associations. Yeah.
Thank you.
Thank you.
And with this, we thank our speaker again.